Thanks to the organizers for giving us a
Hello.
chance to present our work,
Richard
and also
Scheuermann.
thank you
I
all
am
for sticking
the
around
Director
till the
of
end of the session.
Informatics at the
Appreciate
J.
it.
Craig Venter Institute.
Yeah, what I want to
in
tell you
La Jolla,
about is a
California.
method that we've developed
And today I'd like to tell you
identify
about a
informative
machine
features
learning-based
from
method
single-cell RNA
for
sequencing
informative
data.
feature selection
And
that
this
our
work
group has
revolves
developed.
around our interest
Our
in
group has
trying to
been
understand the
especially
cellular
interested in
complexity
trying
of the
to
human
understand the
neocortex, the
cellular
human brain.
complexity
And
of
this
the
is
human
work that we've
brain,
done in
especially
collaboration
the
with the
human
Allen Brain Institute in
neocortex.
Seattle.
And so we've
And
partnered
we're especially
with
interested
Ed
in the
Lean's
human
group at the
brain
Allen Institute
because,
for Brain
or the
Science in
neocortex,
Seattle,
because it's involved in
to
some of the
apply
higher level
single-cell
functions that you find
genomic
in vertebrate
methods
brains,
to try
especially
to
human
understand the
brains,
cellular
like
complexity.
conscious thought
And we're
and
especially
human
interested in
language.
the human neocortex.
And so we're really trying to
because
understand,
the
how does the cellular
neocortex is
complexity
a
contribute
part
to
of
these
higher
complex
vertebrate
brain
brains,
features.
and it's actually involved
And
in
as
we've been
you've heard many
applying
times
single
today,
nucleus RNA
we're using
sequencing
single-cell transcriptional
to
profiling
measure
to
the
try to
transcriptional
characterize the
profiles of
different types of
individual
cells that you
cells
find in the
isolated from the brain.
neocortex. But
And so the
specifically, we're using
workflow
single nucleus
involves
RNA sequencing because if you
generating
try to
a
liberate single
dissection
cells from
from
the
a
brain,
frozen
you
brain
just end up with a
specimen.
bunch of debris
In
because the
some
cells are
cases,
all
we will
interdigitated
microdissect
with
out different
dendrites and
cortical layers
axons.
from the
And so we can float the
neocortex,
nuclei
and
out
then we'll
and get the
isolate
transcriptional
nuclei and
profiles from
perform
nuclei.
RNA
And so,
sequencing
the typical
on
workflow is you
individual
do that, you
nuclei
get
in
your
order
cell
to
by
get
gene expression
gene
matrix,
expression
and then you
data
cluster
from.
that
And
using
so
your favorite
typically what we'll
clustering
do then
algorithm.
is we will
And
use
so this
embedding
is data that
techniques like
we
UMAP or
obtained
TISNE
looking at the middle temporal
to
gyrus,
look
which
at
is
the
this region of the
clusters that are
brain here that's
derived
involved
from
in
this
human language.
analysis in 2
And we
dimensions.
identified
And on this
75
slide, you can
distinct
see on the
cell type
left-hand side that
clusters
there
from that
is
region
a
of
number of
brain.
different clusters that
On
were
the right-hand
identified
side, you can
from
see a heat
this
map
region of
that
the
shows the
brain, the middle
expression
temporal
of
gyrus.
kind of
And in this
prior
case,
knowledge,
the
like
unsupervised
what we
clustering
knew about which
identified
genes were
75
expressed in
distinct
neurons
types
in
of
this region of the
transcriptional
brain.
profiles
So each
that
of
we
the
would then
columns
consider to
represents
be
one of these
distinct cell
75
types.
distinct cell type
So on the
clusters that we
right-hand side,
found,
you see
and then the
a
rows
heat
are
map
these kind of known
and
marker
a
genes.
dendrogram
And the known marker
of
genes kind of
these
tell us a little
75
bit
cell
about,
type
what
clusters.
types of
So
cells
the
at a high level
dendrogram
that we're
actually
dealing with.
captures the
But once you get
relationship
down to the most
between
granular
transcriptional
level, we really didn't
clusters.
have any
So
marker
you
genes
have
that could
cell types
tag
that are
each of these
similar
distinct
to
cell
each
type
other are
clusters that came out
located
of the
next to each other
experiments.
in the dendrogram.
And so what my group wanted
And
to
then below
do
that, we have
is to
the expression
identify
of
marker genes for each of these
well-known
different cell types. And
markers of different
specifically
cell
what we wanted
types.
to do is identify the minimum set of marker genes, because we wanted to use these things for a lot of downstream experiments and
in the
other
human
kinds
brain.
of downstream methods.
So for example, in the top row, we have the
And so
expression
we
that
developed an
It's
algorithm called NS-Forest, which is based on
which
random
stands for
forest machine
necessary and
learning.
sufficient
And again, the idea
random
here is to
forest.
try to identify this minimum
It
set of
involves
necessary and sufficient marker
this
genes
workflow.
that
So
can
we start out with
identify
a
each
cell
of
by
these
gene
distinct cell
expression
type
matrix that
clusters.
comes out of
And so we start out with our
the
input as a cell
RNA-seq
by
data,
gene, a clustered cell by gene
along
expression
with
matrix,
cluster membership
and then
that
we
comes
pass
out of the
that through a
unsupervised
random forest
clustering
algorithm.
analysis.
And basically
And
what
then
we're
we use
doing is
random
trying to build
forest machine
classification
learning
models of each
to
of these
build
distinct cell type
classification
clusters.
models
So
that distinguish
comparing
each cluster
cluster
from
1
all of the other
versus all the other
clusters. So
clusters, and then
the
cluster
first
2
cluster
versus all the other
versus the
clusters,
74
and build
other clusters
classification
that were
models for each
identified
one.
in middle temporal
But rather
gyrus.
than focusing on the
As
models
part of that
themselves, what we're
process,
really interested
we
in
actually
are the
get a
features
ranked
that
list
are
of
used to
informative
build the
features
decision trees,
that
the
are
most
used for
informative features for
classification.
each of these cell type classes.
And we use this
And so we
ranked
keep track of
list
the informative features,
as
and
a
then
way
we
of
realize
identifying
that
these marker
we're getting a
gene
lot of
sets.
quantitative features, and we
We
wanted
also
features that were
added
very
in
binary
an
in their
additional
expression
step to
pattern. We
enrich for
wanted them
genes
to
that
be
have a
expressed at
binary
high levels
expression
in the
pattern.
target
So
cluster, not
they're
expressed at
highly
all in
expressed in the
off-target
target
clusters.
cluster
And so
and
we
not
added in
expressed in
this
off-target
binary scoring
clusters.
function,
And then we use this
which
list
did not
of
reproduce very
informative
well,
features to build single decision trees
to basically
that
re-rank the
include
features
one
based on a
gene,
binary
2 genes,
expression
3
pattern.
genes in
And then
combination.
once we have this
build
kind
single
of
decision
set of
trees, and then we
informative
pick
features,
the
then what we can
combination
do
of
is build
genes
single
that give us
decision trees
maximum
using the
classification
top
accuracy
feature, the
based
top
on
2
the F
features,
beta
3
score
features,
of
4 features, 5
classification
features, whatever.
accuracy.
and
that's how
then
we come
pick
up
the
with this
minimum set
minimum
of
set
features
of
that
marker
give us
genes.
maximum classification
So
accuracy.
on the
Using
next slide
F
here,
beta
you
as
see
our,
a heat map
score.
of the marker
And
genes that
so
were
this is the
selected by
result. This is a
NS4s
heat map
for
now
the
of each of the
75 cell
75
type
clusters
from
in the
middle temporal
columns,
gyrus.
and the rows
So we
are
need
the
about
marker genes that
150
come out of the
marker genes
algorithm.
in total.
So we need about
to distinguish
150
between
marker
these
genes
75
to
different types of
characterize
cells,
the 75 different cell
somewhere
type
between 1 and
clusters,
4
somewhere
marker
between
genes per
1 and
cluster,
4 marker genes.
and the median
Gives
F
us
beta score
maximum
is somewhere
classification
close to
accuracy
0.7,
with a
which is
median
pretty
F
good.
beta score of about 6.
In the box here on the
0.68.
right-hand side, you can see
Two
the
things to
expression
notice
pattern of
about the
these
marker
marker
genes. The
for
first one
one
is
of
in
the
the box there.
clusters.
You can see
And you can
that
see
we
that
need
it's
two marker genes
marker
to
genes that are
characterize
necessary to
that cell type
classify
cluster.
cells from that cluster.
Marker gene is actually expressed in
Cluster.
two different cell
The
type
gene on the
clusters.
top is not only
The
expressed
bottom marker
in
gene
the
is
target
expressed in
cluster,
3,
but
but
also
it's only
expressed
in that
one of
one
the
cell type
off-target
cluster
clusters
where
to the
both of
right.
those genes are expressed
And the second marker
simultaneously.
gene is also
And so it's
expressed
the
in the target
combinatorial
cluster, but
expression of those two
expressed in
markers that
two
gives
other
us the
different
specificity.
off-target clusters.
And then the other
so
thing
the
that's really
combined
interesting from a
expression of these two
biological
marker genes give
perspective is
us
that if you
the
look at a lot of
specificity
the
that we're
marker
looking
genes
for.
for excitatory
One of the
neurons in the
interesting
brain,
things that has come
they're
out of this
long
analysis is
non-coding
that many of the
RNAs.
marker genes
And so
for,
we think that
especially for
there's going to be some
excitatory
really
neurons,
interesting
end
biology
up
that
being
tells us
long
something
non-coding
about what role
RNAs
these
that are
regulatory
annotated as
RNAs might be
LOC.
playing in
And
establishing cell
it'll be
type
very
identity
interesting
of
to figure
neurons
out
in the
why
brain.
it is that
But I think this is
one of
actually the
the
most
pieces
informative
of evidence to
figure
suggest that we're actually
for my
capturing the essence of
presentation.
cell type identity
And that is, so
using
this is the
minimum
original
set of marker
Tisney
genes
embedding that we had
is in this
from the
plot
original
here where we
clustering
generated
result.
a Tisney
What we did
embedding
was we said,
just
okay, well,
using the
let's see what
expression
happens
pattern
when
of
we
the
repeat the
157
embedding
NS4S
just
markers
using
in
this
the middle.
minimum set of marker
And you can
genes.
see that this
And that's what you
closely
see in the middle
recapitulates the embedding
figure.
structure
And if
when
you
we
compare,
use the
the
entire
left-hand
set of
side,
variable
the Tisney
genes
embedding that was
used to
generated
generate
with the
the initial
5,000 most variable
clusters
genes, and the
from the
pattern that you
experiment.
see with just
And so
this
we think
minimum
that this
set of
is
marker genes,
evidence
we're
that suggests
completely
that this minimum set
recapitulating
of
the
marker
embedding
genes is really
structure of
capturing
all
the essence
75
of cell type
clusters
identity
with just this
across these
very
different
minimum
cell
set of
types.
marker genes. So then this is an example of applying this to a different brain region. This is middle temporal gyrus, or sorry, this is primary motor cortex. The interesting thing about that is that we get a similar number of marker genes that we need to characterize the distinct cell types that we see in this different brain region. And so you can use these marker genes for a number of different downstream applications. If you want to do fluorescence in situ hybridization and you want to have a minimum set of probes, you can use this set of markers to be able to look at all these different distinct cell types in spatial transcriptomics data.
For multiplex
we use this as a reduced featured space to do comparative analysis where we can compare two data sets, a reference data set and a new data set.
And you can also use
to see if we're detecting the same types of cells or distinct cell types. And we're also using these marker genes as a way of generating semantic definitions for the cell types that then we can incorporate into reference ontologies.
And so currently we are working on trying to
and finally,
develop a comprehensive
we're starting to work on the entire human and entire
and
mouse
whole
brain in collaboration with
dealing
a
with
group
about
in Karolinska and a group at the Allen
4,000
Institute.
cell types,
And they've defined somewhere between 4,000 and 5,000 distinct types of neurons
we're
in
the process
whole brain
now of
specimens.
using NS4S
and so now we're in the process of trying to define minimum marker genes for each of these distinct cell types.
In the, we have the
here's a
resources
slide
available,
that lists the references, publications,
our scientific publications
GitHub
on the
repos,
development
protocols,
of the NS4S
gene
method,
ontology, or
two
sorry, the
versions
cell
that we have
ontology
worked on so far,
representations.
and their applications.
And
then I'll
most
just
of this
end
work
by
was done by
acknowledging
two of my
collaborators,
Renee Zhang and Brian Averman, and again in collaboration with Ed
I also want to
Lean's group from the
highlight
Allen Institute.
Renee Zhang and
So with
Brian
that,
Averman,
thank
my
you.
two colleagues at JCVI, who have been really instrumental in developing the computational methods that we have
quick
been
question.
using.
There's one here.
Sure.
To this one.
Yeah.
And if you need to skip, you can just click.
Okay, fine.
Hi. So I guess maybe I missed this, but is the random forest fit at the level of individual cells or at the level of clusters?
Clusters. It's at the level of clusters.
Okay. And have you tried methods like Seurat has find variable genes, right? So if you compare that with the top number of genes, like 170 or something genes, how does that work? Or if you use marker genes for each of the clusters, maybe the top 10 marker genes, how does that compare with your method?
Yeah, so we did compare the marker genes that we get from some other methods like Seurat and other methods. And there's some overlap, But what we find is that this set of genes is very stringent, and some of the other methods kind of give marker genes that are maybe not as specific as what we're detecting here. So that's kind of been our experience so far.
Thanks.